Hi, how you going? My name's Mr. Logan. I'm here to talk about diffusion through a membrane, and today we're going to cover the materials, procedure and diagrams for our next lab. So for our materials, we've got dialysis tubing, 2 pieces of string for tying our tube shut. We've got just a splash of starch solution, a splash of glucose solution. Make sure we put them in the appropriate cups, starch and glucose. Then we've got our starch indicator solution, our funnel, and our glass jar of water. And just be careful, you need a very small amount of glucose and starch. I filled the cup just to the bottom of that ridge there, so a very small amount in each cup. So step number 1 is going to take our dialysis tubing and just submerge it and soak it in the water. Let it sit for about a minute, and we want it to soften up and get nice and flexible. After about a minute, you could take that tube out of the water and just try to twist it together with your fingers and allow you to open up one edge of the tube. Okay, and that's where we're going to start pouring our liquids in. So we've got one end open up there, if you could see that. Now with the other side of the tube, the one we haven't opened up yet, we're going to fold it over once like that. Okay, and then we're going to fold it across like this. And now this will be very easy if you have a partner to help you, but take this end and then you're going to just try to tie a simple knot, little overhand knot loop around the edge of the tube. So I've got my loop on there, pull it over, pull both ends tight, and now we've sealed one of the edge of the tube. Put a double knot on there just to be safe, you don't want that coming undone. And we pull it tight. And now we've got one end of the tube sealed closed. Now we can pour our liquids in. Now we're going to take our funnel and put it into the opened end of the tube. Okay, and the first thing we're going to pour in is our glucose solution. Okay, so we filled the tube about a quarter of the ways. You can see that. Next, we're going to add the starch solution, and we only want to fill it halfway because we've got to leave enough room to tie off the top of the tube. So a little bit more starch. Okay, now it's half full, and then we've got to seal up that other end. Okay, so we've got both liquids in there, now we've just got to close it up. So the same way we did the bottom, we're going to take the top of the tube, fold it over, then again fold it across, so 2 folds, and now we're going to pinch that, make another loop. It's a little difficult to do with one hand, but you have your partner to help you out, so it's easier with 2 people in the process. Tie that loop around, seal it up, and then we tie it closed. Okay, so now if we turn our tube over, we want to make sure that both ends are completely leak proof. There's no liquid dripping out of either end. Okay, and the last thing you want to do before putting it back in the jar is we want to just rinse it off in the sink. Make sure there's no starch or glucose on the outside of our tube. Now the tricky part is over, so we're going to just drop our tube into our jar. Okay, so it's completely submerged. And we're going to add starch indicator solution. This is Lugol's or iodine. And we're going to put about 7 drops at first. We want it to get a nice amber color. Okay, so this is a lot of water. I'm going to add 5 more drops just to add more solution. And now you can see we've got our jar of amber colored liquid and our cell inside. So for part of our introduction, I want to go over the initial state diagram, a very important part and something you'll likely see on your end of your Regents exam. So this is basically showing what's inside and what's outside of the cell at the start of the activity, once we put our cell in the water. So just to draw a model, Here we have our cell diagram, and the rest of the diagram represents the surrounding water. So in terms of identifying three substances, what we have inside and outside, we have iodine, glucose, and starch. We're going to represent those with I, G, and S. So on the outside, as far as we know, we've got water, but we also have iodine. So we're going to just fill in a couple of I's to represent molecules surrounding the outside of the cell. And inside of the cell, we have our two other substances, glucose and starch, that we poured into the cell. So we're going to just fill up that model with a couple of G's and S's inside of the diagram to represent the glucose and starch. And we know that because we put it there. Other thing we want to identify here in our initial state diagram is just the color. So remember, on the outside of the cell, we refer to it as that nice amber golden color. So the outside of the cell is amber, and inside of the cell, we haven't seen any change yet, but we'll just say inside is a mix of clear and white, because that starch gives it a little bit of white cloudiness. So inside of the cell is clear and white, outside of the cell is amber. Give it 15 minutes or so and we will observe what changes inside of the cell as well as inside of the beaker.